Epigenetics is a fairly new idea in genetics, and it is a huge growing field when it comes to research, the diagnosing of disease, and has so many implications. So this overview will simply try to answer the question, what is epigenetics? Well, as someone who's a little older, I can attest that epigenetics wasn't understood when I was in school, and when I was in in grad school, it was just beginning to be appreciated. So here's the question. If you were to say compare a normal cell to a cancerous cell and find that there were differences in expression of a certain proteins, ask why is this cell cancerous and the other one isn't, you might say, oh, look, this cancer cell is making cyclin D at a far higher level than a normal cell. Or there might be a protein which normal cells are making that a cancer cell is not. And so the old paradigm would have been, why might that be? It must be a mutation. There must be a change in the genetic code so that this cell has a mutant version that now gets expressed when it shouldn't, or doesn't get expressed, or the protein doesn't work. But that's the cause of this disease. There is a change in the genetic code. But it turns out there's another huge field of possibilities, which then also potentially explains there are so many diseases and disorders which we're still trying to understand. So my reading of the literature on a number of disorders, especially things like, say, autism or schizophrenia, one of the frustrating things is you can then ask, all right, so I see that this gene is linked to say autism in these families, or this gene is implicated here. Here's a mutation which can increase the likelihood of autism. Here's an environmental factor. But what's the main cause of autism? What's the main cause of schizophrenia? And at least my reading of the literature is, well, still working on that. We still don't know. So there's a lot of causes which are known, but what's the major reason? Well, if you're looking for mutations and changes in genetic structure, you may come away with, we still don't understand. What's the major cause? Why does this person have a disease when that person doesn't? What's the cause? Epigenetics offers a new understanding of what seems to be a major cause of disease, right? So
epigenetics refers to the modifications in
it is certainly important. The DNA sequence that you have for a specific gene, and thus then the protein which would be made from that sequence. But another important aspect is, are you using the gene? Because here, if you look at the globin gene cluster, for example, notice we have to unwind this section before the gene can be active. And at one stage in embryonic development, it's unwound and it's used. But then at other stages, other beta globin genes are unwound. So when we consider DNA, its double helix is organized around proteins known as histone proteins. And this can then be organized in a way to form the chromatin, which can be packed to form chromosomes. What determines then the packing there? Well, changes in two different levels primarily. One is it could be a change in nucleotides. So if we look at cytosine, that's one of the 4 nucleotides of DNA, cytosine, you can add a methyl group, that's a CH3 group, to the 5' carbon of cytosine. And this then produces a methyl cytosine, which then has a different effect on the cell than regular old cytosine does. Where does this methyl come from? Well, there is a donor molecule known as SAM, the S-adenosyl methionine, which has a methyl group attached to it. And then an enzyme known as a DNA methyltransferase, or a DNMT. Both of these, SAM and the DNA methyltransferases, they will come up again in this short discussion. These enzymes then transfer the methyl from here to there. And so a methyl can be taken from a donor molecule and then inserted onto a cytosine. Now, we have lots of cytosines, obviously, in the genome, and overall, only about 1% of the cytosines have methyl groups attached to them. However, it turns out that there are what are called dinucleotides. So dinucleotides is just two nucleotides next to each other. And that if a cytosine is next to a guanine, we refer, it's then called CPG, where the P stands for the phosphate. So cytosine phosphate guanine. And there can be regions which, maybe have, a couple hundred nucleotides in length that then have a lot of these. So what is called a CPG island is an area of the genome where you have lots and lots of these cytosines next to guanines. While 1% of regular old cytosines can be methylated, 70 to 90% of the cytosines in these CPG islands can be methylated. So apparently the methylation of cytosine does not apply to cytosine equally throughout the genome, but rather primarily to the cytosines in these CPG islands. How many do we have and where are they? Well, there are tens of thousands of them in the human genome. All right, so there's a lot of these, and they tend to be found just upstream of a gene. They seem to be found in the regulatory areas of a gene, which then determine whether a gene is on or off. So remember, it's not just a question of what genes you have, it's of what genes you're using. Because going back to the cancer example, some cancer cells have all of the same genes as a normal cell. But a gene that should be on is now off, and that's allowing the cancerous growth. And or genes which should be off are now on. Or I apologize if I said the reverse. You get it. So the regulation of genes can be key. So the CPG islands then are important. So when we look at these genes which are determining cell growth or increasing our risk, or affecting our risk of heart disease or asthma or weight gain or diabetes or mental disease or many things, the question is then, let's look at the upstream regulatory areas of these genes. Are they methylated? This will determine whether they are on or off. And very often disease is not, because, you have a change in the order of the amino acids in the protein, but rather upstream of the gene, you have these CPG islands, an excess of these dinucleotides where cytosine is next to guanine, and that their methylation is then affecting whether the gene is on or off. How could it possibly do that? Well, lots of things stick to DNA. And it turns out the things that stick to DNA, whether they be transcription factors or the histone proteins that I'll introduce in the next one, things that stick to DNA can then stick differentially to DNA based on its degree of methylation. So there are proteins which will help to turn on genes, proteins which will help to turn off genes, proteins like histones which affect the packing of the DNA. And whether the DNA is methylated or not affects how these other things bind. So here, for example, we had two identical sequences, but if we remove the methyl groups from the top sequence, this gene is now active and is being used. The bottom gene is not. It is then inactive. So once again, we want to understand disease, how the body works, etc. And then there are just questions, like, once again, just my reading of the literature. What is the major cause of autism and why is its rate increasing? Why is the rate of diabetes increasing or asthma increasing? So maybe it's environmental factors, but it certainly can be genes in that, or not genetic sequences. The change in asthma or autism or diabetes frequency is far too fast for it to be the result of the new frequencies of alleles in the human population. Allele frequency doesn't change that frequency. So it's not the sequences, but it might then be the methylation patterns which are determining the activity of the genes. Now, before we get to the big picture on this one, there is another way that epigenetics can affect gene expression without affecting the sequence of the gene. And that's because DNA gets packed. If you were to go into one of my cells, like a cheek cell, take out its DNA, it would stretch 6 feet long. It would be as tall as me, just about. All right, that's a huge amount of molecule for a microscopic nucleus. But what happens is it gets packed. It gets wrapped around these histone proteins. It gets packed, into solenoids. It gets attached to the DNA scaffolding, or the protein scaffolding for a chromosome. And so we just pack it really tight, and that's how the small amount fits. It fits in such a small space. So the first level of DNA packing is that it gets wrapped around these histone proteins. And one of the things that happens is DNA has a negative charge. The histone proteins have a positive charge. So they're attracted to each other. So it's easy to wrap one around the other. However, you can now attach things to these histone proteins. It could be a methyl group. So you can attach methyl groups to amino acids like arginine and sometimes lysine. And you can attach acetyl groups to lysine. You can attach phosphates to serine and tyrosine. And this then will affect the charges on the histone proteins and the degree to which the DNA is attracted to it. So in other words, if the DNA is highly attracted to the histones, it will more easily tightly wrapped around it. Less attraction means the DNA organization will tend to be looser. And we have enzymes that do this. We have histone acetyl transferases, which can add acetyl groups. We have methyl transferases, which can attach methyl groups. So thus we can change whether the DNA is wrapped around histones by changing, that attraction between the histone proteins and the DNA. If you want to turn on a gene, you loosen this. If you want to turn off a gene, you tighten this. And so once again, we're not talking about a change in the sequence, but rather a change in the activity of the gene. You have genes, but the second question is, are you using them? And these epigenetic changes to the histones, not reflected in any sequence change, is now affecting whether you use these genes or not. And so we can activate genes. There are then also changes. So when we add, say, a methyl group to lysine, which lysine, where in the histone protein, some of these will activate the gene. Some of them will actually turn the gene off. And so it depends on which amino acid in which of the histones we do this to. Now, once again, we can add things like acetyl groups, which could, say, loosen DNA. You could remove acetyl groups, which would then allow you to turn off a gene with histone deacetylases. And so we can regulate gene expression. Okay. Now, here's something a little crazy. Or at least when I was in school, this just didn't seem to make any sense. Some of these changes can be very long lasting, almost permanent. Some of these changes can even maybe affect the next generation. How is that possible? Well, it turns out, let's say we have, we're going to replicate DNA. So a cell is dividing and we have to replicate DNA. So the double-stranded DNA molecule separates it. And now what's going to happen is that new nucleotides get added. But look, here comes an enzyme called DNMT1, DNA methyltransferase 1. It is going to methylate the new strand to the degree to which the parent strand was methylated. So if this was relatively unmethylated, this enzyme would not add much in the way of methyl groups to the CpG islands. But because it's more methylated, methyl groups are going to be added to the new CpG islands on the new strand. Why does that mean? It means that, or what does that mean? It means that if I turned off a gene when I was a child, it could stay off because every time the cell divides, even though I'm getting a new cell with new DNA, it now reflects the epigenetic changes from the original strand. So some decisions that we make, I'm going to turn on this gene, I'm going to turn off this gene, they can become relatively permanent. Now, what I'm about to say is kind of another lecture, so I just want to mention it and not go into it as much. But there is a phenomenon known as imprinting in my cells, and this varies from part of the body to part of the body. Some places, even though I have two copies of a gene, one that my mother gave me, one that my father gave me, in some areas, I might just be using my father's gene. In other areas, I might just be using my mother's gene. So obviously that's important for disease, because if we're saying, oh, here's a recessive mutation, and you're trying to predict its likelihood in the next generation. Well, if there's a certain part of your body where you're only using your dad's copy of the gene, clearly then which version you inherited from your dad, that becomes really important in that case. How is that possible? How can, in a nucleus, you know the difference between the copy of the gene you got from your mother and the copy of the gene that you got from your father? Well, in general, DNA from sperm is more methylated than from ova. And every time cells divide, if they're replicating the original degree of methylation, your cells still know which chromosome 1 you got from your dad and which chromosome 1 you got from your mom, which chromosome 2 you got from your dad, which chromosome 2 you got from your mom. And it might express genes on these two chromosomes differently. This is something that could last your entire life, resulting from the degrees of methylation which were present when you were an embryo. So if changes are made to the methylation of DNA or to the acetylation of histone proteins, they can be long-lasting. There are even studies where the methylation of, say, a female, so for example, let's say this is an experiment done with lab rats. If the females undergo epigenetic changes, it changes their methylation. This then affects the DNA they pass on to the next generation, which could affect gene expression in the next generation. So there is no change in the DNA. The DNA sequence is exactly the same, but there would be an increased likelihood of disease because of the likelihood of the genes being turned on. This is epigenetic. And I'd like to just kind of end because as we study disease, let's say asthma, for example, we're asking in the asthmatic, sure, there are genes whose mutations predispose you to asthma. We know that. There are environmental factors which could predispose you to asthma. We know that. But another aspect which we are just beginning to appreciate is that environmental factors might then cause epigenetic changes, which then result in this. So for example, things like cigarette smoking, air pollution, maternal smoking, air pollution exposure. So a woman, if she smokes when she was pregnant, could actually then change methylation of DNA, which would then get passed down to her offspring, which would then affect gene regulation there. So there are environmental factors which can change them, have epigenetic changes on relevant genes, and they've been identified. So these are genes whose activity are relevant to asthma, and it can be, if you ask why in the asthmatic is this gene on at a higher level than in most, or why is this gene off, etc. Why is this asthmatic using lymphocytes more in the Th2 pathway than the regular Th1 pathway, etc. Why is the asthmatic different? It might be because of something that happened long ago when they were a child, something that happened during fetal development, which cause epigenetic changes, which then just continues to replicate itself each generation, since the genes which are turned off are then modified after the S phase of cell division, so that the same level of being activated or inactivated applies. So this, it turns out, when it comes to weight gain, diabetes, these genes having a role in type 1 diabetes, we know that there are genes involved. We know that there are environmental factors involved. But it turns out that some of the genes which are involved could be involved not because of a mutation, but because of an epigenetic change, which then affects the expression of these genes, to what degree they are used. And then there are things such as not only say weight gain and nutrition in your life, but even when you were developing as a fetus, your mother's level of nutrition, her weight gain, your weight gain as a fetus or early in life, these could then have effects long lasting, because every time your cells divide, there were genes which were turned on or off early in life, or even in fetal development, which are now more likely to be turned on or off for the rest of your life. Now, last couple of points is, here's a chemical, all right, which we can be exposed to, which can change levels of methylation. So as we start asking, what could cause these epigenetic changes, which could now cause long-lasting changes in gene expression? Well, it turns out there are things in our diet. All right, so remember that SAM is the donor molecule, but how much SAM is in your body can be affected by your diet, the alcohol you drink, but also folates, which are mandatory things added to our diet and put into maternal vitamins. And so so here's a question: to what degree is your diet affecting the methylation of genes, which affects gene expression? There are lots of pharmaceuticals which are known to cause epigenetic changes. So if you take a medication for, disorder 1 or whatever, does that cause epigenetic changes, which will change your gene expression, which later in life, makes you more or less likely to develop, disorder 2. And I say that, I have an appreciation for this because, one, when I was in school, this wasn't known. And once it started to become known, at first I was like, how would that even work? I don't get it. I don't believe this research, I just don't get it. And it turns out there was this huge aspect of how genes are regulated that wasn't appreciated. And so now, as we try to understand cancer, we have to in addition to looking for mutations, look for, what are the changes in methylation, for example. So in general, you can say this gene, which we need to stop cancer, so P53, okay, that's the major protein which prevents cancer. But in this cancer patient, it's hypermethylated, so therefore it's more likely to be turned off. And so the individual has the gene that would stop cancer. They're just not using it because of those epigenetic changes. There are epigenetic changes to other genes as well, like retinoblastoma, RAS, P15, which say this cancer patient is now expressing the proteins at a different level from the non-cancer patient because of epigenetic changes. In addition to specific methylation changes to one gene, so to this gene or to that gene, sometimes we see what's called a global or a general change in methylation. So there could be a global hypomethylation. I'm sorry, there should be a Y. Or a global hypermethylation, where just in general, an individual has more or less methyl groups attached to their DNA as normal. So sometimes global methylation is linked to cancer. So now more inappropriate genes are more likely to be turned on. I've been focusing on the methylation of DNA, but also cancer can involve epigenetic changes to histone proteins. And so there have been a number of cancers where it's not a mutation that's linked to the cancer, but the epigenetic changes which are changing gene expression. And then it turns out this seems to be involved in lots of issues. So as we age, why is my body just not working as well as it once did? Why am I not healing the way I did, et cetera? Well, in aging, there seems to be, a global hypomethylation, but also then specifically the hypermethylation of specific genes. When we ask, what's happening in the brain that is affecting this aspect of personality? Well, epigenetic changes have been linked to a number of disorders, and then Alzheimer's and other phenomena. We know from the studies of mice that methylation can be changed by the amount of maternal care that infant mice get. And so as we consider how children grow up, there is the possibility that some children do not have the same benefits as others when it comes to, stability in the home, parental care, nutrition, exposure to environmental toxins, et cetera. This could potentially change epigenetically aspects of the genes, which will now affect that child's incidence of disease for the rest of their lives. And once again, this is a huge area in biology. So if you're going into, say, genetics, you want to understand this because, this is an active area of research as we look into, say, heart disease, asthma, all of these things. Just a couple of finer points. Men and women, they don't vary a lot genetically. I mean, men have all of the genes that women have. Except they only have one copy of the X-linked genes, and women have all of the genes that men have, except the SRY gene on the Y chromosome. But it turns out that methylation patterns are different, and some of the reporting or some of the papers I've read, I mean, they're different to a greater degree than one would expect. So surprising, how different. And so, for example, if you're looking at, the gender difference in, a certain disease incidence where this disease seems to affect women more or men more, it might be differing levels of methylation, which are involved. And so this has so many applications. Just to end with like a simple one. A lot of people will ask, well, monozygotic twins, they have all of the same DNA. Well, why can I tell them apart? Why does this one have more freckles than that one, for example? So, their DNA is the same, but, I can still depict some differences. Well, it might be these epigenetic changes, which happened after conception, which they have the same genes, they're just using them to different degrees. So if you're studying biology and perhaps going into genetics, as this is part of my genetics course that I teach, this is a fairly new area of biology. Not only do we care about the sequences of the genes, but then the epigenetic changes. Is the DNA being wrapped around the histones, which tend to turn it off and make it less active? Is the DNA methylated when methyl groups are put on the cytosines? This is more likely to turn a gene off. These are affecting whether you're using a gene. And so many aspects of our physiology, so many diseases are not because of the sequence of a gene, but rather the degree to its expression. So epigenetics is, critically important, and also a very active area of new research because there's so much here apparently, which we are just beginning to learn.